Hello, this video is about the Algae facilities and kinetic model platform that our team have. At first, we're gonna introduce the Algae cultivation facilities for bench and the pilot scale study. In Washington University, we have 30 and 37 degrees C space for algae cultivation. The shakers are located in each room, and the light and electricity are equipped and the intensity can be adjusted. Only sterilized cultures are allowed in the room, and the room is cleaned every day. In Washington University, there is a greenhouse named Jenny Goldfarb plant grow facility, and there are a lot of plants growing in the greenhouse. The temperature is stable with about 80 degrees Fahrenheit. This space is suitable for algae study by their several years of experiences, and the open tank system can be applied for the scale-up study. With the continuous water circulation, the culture can be well mixed and the fluctuating light condition can be mimicked as well. Also, there are various algae cultures stored in the Lincoln University for our study. Because of the agriculture-focused research interest of the Lincoln University, there is also a greenhouse for the algae cultivation. And Lincoln University has the liquid chromatography, mass spectroscopy, and the gas chromatography facility. And next is about the photobioreactor for the algae cultivation.
Hi everyone, today I'm showing our facility to perform autotrophic and hydrotrophic co-culture experiments for the DOE project. Here is a picture of a photobioreactor we use. It is a multi-cultivator MC1000OD. It contains 8 separate columns to grow light-driven microbial cultures. The temperature is controlled universally for the 8 columns. The air bubbling rate and light intensity can be controlled individually for each column. The photobioreactor also has optical density measurement and temperature monitor that can report real-time cell density and temperature controls. Here is a closer look of the reactor columns with growing cyanobacteria cultures. And this short clip is showing a co-culture system where the light-driven algae strain is growing in the liquid culture, and the hydrotrophic bacterial strain is immobilized in the spherical hydrogels. Thank you.
And our last part is about the algae system kinetic model simulation tool, which is called Magma.
Hello, and welcome to a demo for the microalgae growth modeling application, nicknamed Magma. The purpose of this application is to create a platform for the efficient creation of kinetic growth models for microalgae cultivation systems that is easy to learn and use without the requirement of extensive MATLAB knowledge. The goal is to allow users to either create their own modeling schemes or to make modifications to an existing scheme created by other users. Creating a model from scratch allows for vast flexibility and specificity in the model, while modifying an existing model gives quick access to a defined model structure and parameters. This demo will give you a quick overview of how to build a model from scratch. Along the top of the application window, there will appear a set of tabs. The first tab is the Home tab, where the functionality to load existing modeling schemes will eventually be implemented. The Environment tab is the beginning of the model building process. It allows the user to define environmental conditions for the model, such as incident light and temperature. Modifying these conditions is done by selecting a condition from the list and clicking into the a value field and the unit field and setting the desired values and clicking update. These functions can be defined as a constant or as a function of time, depending on which parameters or conditions that's applicable for. The Species and Chemicals tab allows the user to specify which biological and chemical species are involved in the model, along with their initial concentrations. The chemical species include any nutrients, growth inhibitors, and metabolic products. The biological and chemical species are specified by selecting the species from the preset list and setting the initial concentration. If a species is not in the preset list, it can be inputted as a custom species. The Interaction Models tab allows the user to specify individual relationships a chemical has with a biological species. The user will define the chemical substrate, the relationship the substrate has with the biological species, and the biological species. The user is then able to build a function representing the individual rate of change relationship of the concentration for the biological and chemical species selected. The functions built by the user are in the form of an f and a g function. For a given species, all of the f and g functions will be compiled according to the formula to create the full rate of change equation, and same for the given chemical. The formula are displayed here for the biological species and here for the chemical species. These individual relationship functions will ultimately have the units of 1 over time, as they will be scaled to concentration per time, as shown here for biological species and here for chemical species. The purpose of this method is to provide the user with flexibility in modeling while maintaining an organized structure. For example, a basic monod relationship can be established between nitrate and 273. Clicking the update button will save this function as the function representing the relationship of nitrate being a nutrient for 2973. This function utilizes the nitrate concentration, a dependent variable of the system that is listed in the system variables table here, where other system variables such as time, culture, volume, temperature, etc. are listed for the user to use in their functions. The Growth Parameters tab allows the user to specify the value and units for any parameters that are used in the rate of change functions that aren't independent or dependent variables of the system. For example, the half saturation constant for nitrate for 2973 can be set to a value of 0.15 grams per liter. Now we can also rename the parameter to keep track of everything in the system. So we can call this the half saturation constant for nitrate, and they will update as shown here. When all relevant individual relationships and parameters have been specified, Magma will compile these relationships into an aggregate ODE system to represent the model. The ODEs in this system will be displayed on the Model Summary tab. This portion of the model is still in development, but it will look something like this. The user will then be able to view the compiled functions and go back to previous tabs to edit the relationships if necessary. The user can then go to the Plot Customization tab to specify which plots they want to create. This part of the user interface has not yet been created, but it will give the user some options for plots they want to create, such as cell concentration over time, nutrient concentration over time, final cell concentrations, etc. After selecting and customizing that plotting output, the user can then view the plots on the Plot Display tab. Thank you.